Information and Communication Technology presents Educational Satellite Television Programs.
Hello, teacher. Hello, students. Welcome to today's lesson. In our last lesson, we learned about the methods of controlling microorganisms using heat. We also learned about aseptic technique, inoculation, and incubation. In today's lesson, we will learn about methods of growing microorganisms and staining. Let us get started. Scientists often breed large numbers of microorganisms in the form of colonies in a lab. This is called culturing microorganisms. Scientists do this because it allows them to learn more about the cells. They can see what microorganisms look like, how they function, and how they are killed. To culture microorganisms, everything they need to survive must be provided. They grow fast in warm places with a continuous supply of oxygen. They also need ions, proteins, minerals, vitamins, and carbohydrates for energy. A liquid or gel, called a culture medium, is designed to support the growth of microorganisms. There are different types of culture media for growing different types of cells. But the most common culture media for microorganisms are agar media and nutrient broths. Agar is a substance that dissolves in hot water and sets to form a jelly. When agar is combined with all the right nutrients for the microorganisms, it is poured into a petri dish and left to cool and set. After it sets, the cells can be placed on the agar and begin to culture on the gelled surface. The other way to provide nutrients to microorganisms is through a nutrient broth in a culture flask. Unlike agar, which gels, the nutrient broth will culture the microorganisms in a liquid. Another important requirement for microorganisms to grow is having a clean environment. All the equipment, like petri dishes, containers, and the culture medium that is used to grow microorganisms need to be sterilized. Small amounts of other unwanted microorganisms on these materials could contaminate the new growing cells. Normally, the process known as autoclave is used to sterilize the materials. As we learned in the previous lesson, autoclaving uses high-pressure steam to sterilize. For our first activity, students, write down at least 2 requirements needed for microorganisms to grow.
Students, let's get ready. Begin. Time's up. Let's get back to our lesson.
Welcome back, students. There are a few factors that are important when scientists grow microorganisms. Some of the factors that we have learned about are a warm environment, plenty of oxygen, sterilized petri dish, container, and surroundings by using heat sources, hot air ovens, or chemicals, carbohydrates, ions, proteins, and minerals. These can be in the form of a culture medium. If you listed any of these requirements, then you answered correctly. To grow microorganisms, there must be a series of steps that are followed to make sure it is done successfully. Let us go through the steps. First, sterilization of the area and the utensils must be done. To sterilize the area, chemicals or heat sources such as dry heat or hot air oven must be used. The tool used to transfer the microorganisms to the agar, which is called the inoculating loop, must be sterilized. This is done by holding it over a flame and then letting it cool. The next step is to dip the inoculating loop into a sample of the bacteria or microorganism you want to grow, and then make zigzag streaks with it across the surface of the agar. It is important to work quickly and efficiently in this step because the microorganism is exposed to the air. The final step is to secure the lid of the Petri dish to prevent microorganisms from the air contaminating the culture or any of the organisms from within escaping. Let us watch this video clip on growing microorganisms.
To grow bacteria, you just need agar and Petri dishes. Melt the agar in a hot water bath or microwave, swirling frequently to distribute the heat. Open the lid of the Petri dish just enough to pour some liquid agar in. Tilt the dish until the agar covers the whole dish, then let it sit for an hour so the agar solidifies. Use a sterile swab to collect bacteria from a cell phone, kitchen sink, or any other surface you want to test. Lightly rub the swab across the surface of the agar in a zigzag pattern, turning the dish a few times for maximum coverage. Label each dish and put them in a dark, room temperature place. In just 3 to 7 days, you should see small round bacteria colonies growing on the agar. Different kinds of colonies will have different colors and textures.
Welcome back, students. The next topic we will be learning about is staining. Scientists use a technique called staining that colors the microorganisms so they can be seen better. Some stains make it easier to see the microorganism, while other stains allow the scientist to observe certain functions within the organism's cellular structures. For example, crystal violet is a common simple stain that turns all bacteria purple. By staining, scientists can see the shape of the bacteria. Staining helps scientists to identify both a cell's shape and cellular structure, but it can also help to see various kinds of cells in a culture. Different cells have different cell walls. The cell walls determine how much stain the cell will absorb. This is called Gram stain. For example, When chemicals are added to crystal violet and applied to a culture of bacteria, the various types of cells will absorb different amounts of the stain. When a scientist looks at the culture after the Gram stain is applied, he will be able to identify all the kinds of cells in the culture. Here are the steps for the Gram stain procedure. First, crystal violet is applied to the slide with the bacteria sample. After a minute, rinse the slide with water. Apply iodine to the slide. Iodine binds with crystal violet and is then unable to exit the cell wall. Rinse the slide with water after a minute. Next, apply acetone alcohol to the slide. Alcohol will remove the stain from the Gram-negative cells. After 10 to 15 seconds, rinse the slide with water. Apply safranine, which is a counter stain, to the slide. Rinse after a minute. Finally, blot the slide dry. It is now ready to be viewed under a microscope. For our last activity, students, write a sentence to explain why the process of staining is useful to scientists. When you have completed writing your sentence, share your response with the student sitting next to you.
Students, let's get ready. Begin. Time's up. Let's get back to our lesson.
Welcome back, students. Did you share your response with your classmates? Staining is essential for scientists to use when they are growing microorganisms because the different colors of the cells make it easier to see how they look and how they are structured. Staining helps the scientist to distinguish between different kinds of cells within one culture. In this lesson, we learned about the methods and requirements of growing microorganisms. We also learned about the process of staining. I hope you enjoyed today's lesson. Until next time, thank you, students. Thank you, teacher.